Hi everybody, Noah here with Learn Meta-Analysis, and this is going to be a really fun video. We're going to talk about outliers and influence, which is something that not a lot of people actually talk about in meta-analysis, and it makes me so sad deep down inside as a total nerd because I feel like this is something incredibly important to the meta-analysis process, and people just don't really talk about it that much. Even when you read the meta-analyses that are published, a lot of times they don't talk about if they even searched for outliers or how they examined influence. So let's work towards best practices here and let's check out what these things mean and what we can do about it depending upon what we find. So by the end of this video, I want you to be able to identify various schools of thought on what to do with outliers and what constitutes an outlier. And I also want you to be able to interpret outlier and influence statistics from metaphor output. So first question that comes to mind, what is an outlier and what on earth do we do with them? Well, here's where part of the problem itself lies, okay? I found a variety of definitions of what an outlier is, but very few were for meta-analysis specifically. So what does that mean? Well, that means that outliers is kind of a squishy construct, and it can mean a lot of different things to a lot of different people. But what I found is it's often not discussed in papers. Sometimes you do see people acknowledge it, but I would say of the meta-analyses I can remember reading, very few actually talk about outliers. So what I feel is the more important question, because I feel that you can rationalize using many different definitions of outliers as long as you have a good rationale for doing so. But my question that I always wonder about is, what do you actually do with an outlier? So let's say you find an outlier. What do you do? Well, believe it or not, there's actually a lot of different perspectives on this as well. So I would say most people agree that you should, at the very least, re-examine the study. Okay, and when you're doing this, you're checking to see if you had data entry errors or if the study is notably different than others in your analysis. Now, what happens if there's no data entry errors and the study's not notably different? Well, some people like to change or adjust the effect size to slightly larger or slightly smaller than the next closest effect size. The question I've always had is by how much? And I haven't found any explanation for that. Most of the time people just say we adjusted it, but it doesn't say how much they adjusted it or why they adjusted that. So another approach is to actually remove the effect size from the analysis. I've seen this done too, but what if it's a legitimate study, right? What if this was actually a well-done study? There's no data entry errors anywhere that we can see, and they actually legitimately found this. You're just going to remove it because it becomes an outlier by whatever definition you have? That's always been a little bit problematic in my brain. So here's another question, or another option, I should say. If you check the study, there's no errors, the study isn't notably different, it seems like it was well done. Another option, and this is the option that I tend to prefer as of now in 2024, is to ignore the outlier status and check for influence. Now, when I say ignore, I do report this. I do say this was, we identified this number of outliers, and then we checked for influence. So when I say ignore, I don't mean ignore it when reporting your paper. You should definitely report it in your paper. But when I say ignore it, I mean I don't change the effect size unless, I don't change the effect size at this point. I then look for influence. So let's talk a little bit about study influence, because this is something I don't see talked about nearly enough. So study influence tests or diagnostics are helping you examine if a particular effect size has a significant effect on the overall meta-analysis. This would be really good to know, wouldn't it? Like, let's say you're meta-analyzing 15 studies, and let's say one of them is actually really influential. Isn't that something you'd want to know? Yeah, so why don't we talk about it more? People should be talking about this. Such a cool statistic set that we can work with here. So here's some common metrics that I've seen. So first we have a Cook's distance, which is essentially the distance between the data set with or without the respective study. We have DF betas, which is the number of standard deviations the overall effect size changes if you exclude each respective study. And we have a hat value, which is a little bit more complex to explain, so I just want you to know that it's a measure of influence. And again, if you're going to actually be running meta-analyses, you should dive into each one of these things and understand what they actually mean a little bit more in depth, partially just so you know it, but also so that way you can talk more informatively about it and make better statistical decisions as you're actually running your analysis. But anyway, for readers of meta-analyses, these are three common metrics: Cook's distance, DF betas, and the hat value. So the next question, what do we do if a study is influential? Well, again, we're going to examine the results with and without the study present, right? We want to make sure that we are transparent in our findings. The other thing that I'll mention here is just because a study is influential doesn't mean it's bad. Okay, this is really a descriptive information for us to use. So I ran a meta-analysis recently where I found one study was very influential. Guess what? That study had a much larger sample size than any of the other studies in there. So it makes sense that that study was more influential than others, right? So just because something is influential doesn't mean it's bad, doesn't mean it's a negative thing. We need to take in this information and contextualize it within the context of the analysis that we're doing. Okay, so let's look at an example of what some output might look like from metaphor. And I know this seems scary, like when you see all these numbers on the screen, and then we can see all these things like R student and DF fits and cook.d, like what does all this craziness mean? Here's the thing. Metaphor output for conventional models is actually very, very easy to interpret. And that is because in the final column called inf, which you can see all the way on the right, we're looking for an asterisk. Okay, if there is an asterisk in the final column, that means that study should be checked out because it has it's either a potential outlier or has significant influence according to one of these metrics. So I include the web link here to more information about this process and what this analysis is actually doing. This is using the influence function from metaphor. But what I want you to have as a takeaway point here is if you're running a conventional model, it's not like you need to do some really, really complex process to be able to check out outliers and influence. It's actually very, very simple if you're using R. If you use my software Simple Meta-Analysis, I built this into that as well, so it's very easy. If you're using some other graphic user interface-based programs, they don't, at least to my knowledge, I can't think of any others that automatically will flag these outliers and influence for you. So you have to go about doing it a different way, which is perhaps why we don't see it reported too broadly. But hopefully some of these free software options will help make this more accessible, and we'll start seeing this reported more into the future. So 3-level models are a little bit different. We don't have that influence function directly that's going to give us the same sort of output. So what you see here is a spreadsheet that was generated by my Simple Meta-Analysis software. This is it's based off metaphor. So we're looking at Cook's distance, DF betas, and hat values specifically. I have 2 links and papers down here that I think you should read if you're actually conducting meta-analyses. The first is some reference information about influence diagnostics for RMA.MV models, and the second is a paper I just think you'll find helpful. So with Cook's distance, we are looking for, I guess I should start with outliers since that's the first column that we can see here with outliers. So we're using Van Liese's definition of an outlier, which in this case is actually looking for effect sizes that do not have confidence intervals that overlap the overall meta-analytic confidence interval. So I know that sounds convoluted, but as you can see in column C, it just marks it true if it meets that definition, okay? Then if we look at column E, that says Cook's flag. If that is marked true, then that means it has significant influence according to Cook's distance. And with Cook's distance, we're looking for values over 0.5. Next, we can see DF betas. We have DF betas underscore flag. That's column G. Here again, we are looking for values that are either less than -1 or more than +1. And it's going to mark it true if it meets that requirement. And then last but not least, we have hat values, and the formula is listed here on the screen. I'm not going to repeat it out loud, but it will essentially automatically do those computations and calculate if you have a problem there too. So when we look at this output from Simple Meta-Analysis, you can see that if we look at this, for example, this second comparison from Kim 2009, it's an outlier and it has significant influence according to 2 of the 3 metrics. So we would definitely want to go back and take a look and make sure, hey, were there any data entry errors, or is this experiment notably different than anything else in our sample, or what might explain why this is an outlier and why it has significant influence on our sample, and make our decisions from there. So to summarize, we really need to be checking for outliers and influence to ensure our findings are not being skewed for reasons that we're otherwise unaware of. It really bothers me a lot that we don't see outliers and influence talked about more in current papers. I am guessing this is just because it's not necessarily well known how to calculate these things. For our users, we see these things calculate a lot because, as I showed you, these are features that are built into Metaphor, and a lot of meta-analysts use Metaphor to run their meta-analyses, so it's very simple to check for outliers and influence with that. But if you're using a lot of the graphic user interface programs, outside of Simple Meta-Analysis, the one I've developed, which is built on top of Metaphor, I don't know of any off the top of my head that actually do this. So I think that's why we're not seeing outliers and influence reported very much. I really hope this is going to change. So what do we do if a study is an outlier but not influential? My personal opinion, if it's an outlier but not influential, I leave it be. because I feel that if it's not a data entry error and it's not notably different than the other studies in my analysis, that is what I would consider to be a legitimate effect size, and I'm not going to remove it from my analysis simply because it came up under one definition of an outlier, especially when we can't even agree what outliers are. So, what if the study is influential but not an outlier? In many cases, I'd leave this be as well, assuming that when I checked, there were no data entry errors and it wasn't notably different than the others in the analysis. There may be a reason why this study is influential. Perhaps the sample size is notably larger than the others. We need to consider this contextual information before we change anything. Okay, so what do we do if a study is an outlier and is also influential? Here's what I would recommend. First and foremost, examining the data for any data entry errors. This is what I do regardless of if a study is an outlier or if it's influential individually. I always go to this first because data entry errors happen. We're human. It happens. We make mistakes. We fix them, and then we move on. Second, I examine the study itself, and I ask the question of, is there anything notably different about this study than others in the sample? Okay? So if there is, you need to ask yourself if you want to retain the study in your sample or not. And if there isn't, is there a reason to actually remove or edit this effect size? These are decisions only you can make. So overall, here's my opinion. If the primary studies are sound and consistent with the rest of my sample, I'm really hesitant to change any effect sizes or to remove them from my data set. In my opinion, the overall idea of most meta-analyses is to try and understand the entire field. And if I am arbitrarily changing things or removing things, I do not feel that I am getting that comprehensive coverage of the field that I want. So that is my personal opinion on this right now. It may change with time, but that's where I stand now in 2024. So I hope this has been a helpful video for you learning about outliers and influence and meta-analysis. If you are still interested in this, be sure to read some of those really those links that I provided and those extra papers that I provided as we go through here. Really, really great reading for anyone interested in meta-analysis. So please do take the time to read them. So thank you guys. I'll see you in the next video.